Welcome to our third of 5 fully vodcasted lectures that we'll have this semester. And if you are watching this from your regularly scheduled classroom location, then it will be Monday, the 5th of March. And you'll be gearing up to turn in your homework assignment tomorrow, which is due on the 6th. To help you out with that, even though I'm not physically there, I'll be there tonight on Illuminate from 7:30 to 8:30. So if you need any help with the homework, feel free to come and find me there. I will be taking a break from waxing lots of skis to come and visit with you. I'll also hold a Tuesday night office hour rather than on Second Life. I'll hold it on Illuminate again, and I'll do that again from 7:30 to 8:30 p.m. So if you need to find me for help on any of the homework, I will be there with bells on. And I wanted to make one quick note regarding lab. Tomorrow you'll be doing lab 15, and it's the cornerstone lab for all of the unknown identification procedures that we'll be doing soon. And so help your TAs out because they'll have been working very hard to prepare a good lecture to get you ready for all of this. But if you can help them out too by being prepared for Lab 15 and kind of getting on the up and up with what's going to be happening that day, it will really be super, super helpful to your TAs, and I will be forever thankful. Okay, when we left off last time, we had finished our coverage of transcription, and we had just begun talking about translation. We're going to move from there in reminding ourselves that we had just talked about the way in which a polypeptide chain is built during translation, where the new amino acids are constantly added to the carboxyl terminus of the ever elongating chain. Now I've gone ahead and drawn a really cheesy drawing. I was having trouble drawing sideways, but this gives you the idea of what we were talking about last time, where the mRNA is read by the ribosome that I've shown in green there, with its two subunits, and we'll be talking more about that today. So that's the ribosome. And as the ribosome reads down the mRNA, it converts the mRNA code into polypeptide chain. And so the carboxyl terminus is always inside of the ribosome, the 50S subunit, where it's getting new amino acids added onto it, which means that the first terminus to appear out of the top of the ribosome is actually the amino terminus. So this helps with a little bit of a visual of what's going on there. But we all know that mRNA has in it the 4 different nucleotides, and those are G, C, U, and A, and those obviously can't individually encode for each of the 20 amino acids, right? The math doesn't add up, does it, Greg? Because we see that there are 4 nucleotides, but there are 20 amino acids. So you might think, oh, well, okay, what if those nucleotides are read 2 at a time? So there are 4 of them, and they're read 2 at a time. Okay, well, that would give us 16 possible combinations. But that still doesn't do the trick, does it? Because we know that there are 20 amino acids. So in order for an mRNA to encode for the polypeptide chain, it's going to have to be read 3 nucleotides at a time. So that is 4 cubed is what's going to give us enough combinations to encode for 20 amino acids. And in fact, maybe you're laughing and thinking, yeah, I guess it's enough, Rachel. It's a heck of a lot. So we're going to see there are extras. So let's write a bit of this down. During translation, amino acids are added to the carboxyl terminus of a constantly elongating polypeptide chain. Each amino acid is going to be specified by a 3 nucleotide sequence. And I bet by now you're screaming at the vodcast and saying, yeah, duh, Rachel, this is the codon. Well, the name that we give to the 3 nucleotide sequence that specifies an amino acid during translation is a codon. So codons specify amino acids. Let's go ahead and write down that combination of nucleotides that allows for enough possible combinations. We know there are 3 nucleotides per codon, and we know that there are 4 nucleotides total in an RNA. So 4 raised up to the 3rd is going to give us way enough, 64 different possible codons. So this is kind of strange. Right now you're probably kind of frantically writing this down, but maybe you're also scratching your head because 64 is far greater than the 20 that are needed to specify those amino acids that we see recurring in proteins. So this means something, right? This means that there must be the possibility for more than one codon to specify a single amino acid. And that is very true. It's also true that there are several different codons that are just utilized as stop codons. That is, rather than signaling an amino acid at all, they simply signal, whoa, stop that ribosome, right? Hold it back. And that'll give us 3, actually, that are used to specify stop. So that still leaves us, however, with 61. So UAA, UAG, and UGA, those are our stop codons. They don't specify any amino acids at all. But that still leaves us with 61 codons that do specify particular amino acids. So what that indicates is that it must be true that several different codons can specify the same amino acid. And this is something that it's a very sexy word, actually, degeneracy. Degeneracy means that within the genetic code, we can see variances within a codon and still see that it encodes the same amino acid. That's called degeneracy. More than one codon specifying the same amino acid. Below on this, you see the table for codon usage. And this is a pretty sweet table because it shows you all about degeneracy. That is, it tells you exactly what codons specify what amino acids. Now, don't worry, I don't want you to at all memorize this table. It's something that you would always have accessible for you to use. But this table is really important because it allows us to it makes it so that we can be good ribosomes, right? So that we can understand what amino acid would be incorporated as a ribosome is reading across the mRNA. So let's look at how we see not only the stop codons on this table, and let's go ahead and bring those up. UAA, UAG, and UGA, all of these specifying stop. But we also see a very special codon that specifies start. This is the AUG codon. So AUG always is it's like the landing pad where all of translation begins. It's what specifies the beginning for any synthesis of a polypeptide chain. So AUG, it kicks it all off. Now, in prokaryotic cells, AUG specifies a special amino acid called F-met or N-formyl methionine. So let's write that down so that we know that within prokaryotic cells, this AUG not only specifies a simple methionine, in eukaryotes it's just methionine, but in prokaryotic cells it specifies F-met. Now, this is a formulated derivative of methionine, so it is methionine, our group's the same and everything, but it does on the amino terminus, it has a formal group. It's just slightly modified is all. So that's always going to be what kicks off the polypeptide chain in any given prokaryotic cell. So the first amino acid, if you will, that you see peeking out of the top of a ribosome is always going to be F-met, if we're talking about an E. coli cell or any other prokaryote. So F-met, the start amino acid, signified by the AUG start codon. Now we also see the stop codons, but now we need to talk about some of the other codons, which clearly have a lot of degeneracy. So notice right here that all 4 of these particular codons, CUU, CUC, CUA, CUG, all of them specify leucine. We see more than just that being degenerate. So we could go down here and see all 4 of these codons, GCU, GCC, GCA, and GCG, all specifying alanine. Lots of degeneracy, right? So let's write down a few others here to keep us in breathless anticipation. So we've got UCU, UCC, UCA, and UCG all specifying serine. So we can see again more degeneracy. Now, another thing that we can notice is that sometimes not all 4 of these similar codons will specify the same amino acid, but they might specify very highly related amino acids. For example, see GAU and GAC both specify aspartate, but notice that their similar codons specify another very similarly structured acidic amino acid, glutamate, right? So we recognize that sometimes similar codons might specify not the exact same amino acid, but very highly related amino acids. So we tend to also notice that in certain cases there are similarities between amino acids and their second positions of the codon. So for example, if we look at our aromatic amino acids, so let's see if we can hunt down some of those, such as phenylalanine and tyrosine. Sometimes you'll see similarities between their codons. So you can recognize some of those similarities here, like U and C stay the same, and UU, but only the second position seems to change between tyrosine and phenylalanine. So lots of cool stuff that you can see on the codon usage table, but maybe more importantly than anything is to know how to use the codon usage table. So let's look at how that works. I'll pop out a highlighter so that we can better see it all happening. Over here on the left, this gives us the first position. So if you know you have a codon that begins with a C, well then you would start there. Now the second position is given by the top row. So if the second position is also C, you would go there next. But if you know the third position finally is another C, then you could put it all together. C, C, and then finally C, right? We would come here at proline. So that's how that works. Find the first position, then the second, then the third, and it tells you what amino acid it specifies. So let's practice just a little bit. Tell me what amino acid is specified by the codon GGA. GGA. Okay, so I'll erase while you're finding it. Hopefully you're figuring it out. G. Go to the second position for G, okay, and then A would give you glycine. So GGA specifies glycine, right? So we're getting the hang of how to use the codon usage table, hopefully. And at this point, we can talk a little bit about how not only does AUG specify FMET and the start codon, but AUG is super important because it also specifies the entire reading frame. So that means that when a ribosome binds the mRNA, it's not going to kick things off until it hits an AUG. It's like it has sort of a lead-in before it begins. And we know that, right? Because we know there's that non-translated leader sequence. So let's look at how AUG will set the reading frame and always dictate what kind of triplicates will follow it. So looking at, let's write it down. So the mRNA also carries information that indicates where the coding region actually begins. That is, AUG sets that the way in which the triplicates will be read. It sets the reading frame. So when we look at this sample mRNA down here, we could, if we were very bad ribosomes, we could begin reading it in triplicates by reading at the very beginning here and saying CGA, UCA, UGC, but that would make us really bad ribosomes and we would screw up the entire reading frame. That is, the entire reading frame would be off. So we have to remember that transcription or translation only ever begins at the AUG start codon. So you would never set the reading frame prior to hitting that AUG start codon. So let's do this right and make better ribosomes and find the AUG start codon before we begin. So scanning down, nope, there it is. AUG sets the reading frame. So now all of the codons are read downstream of the AUG. So our second codon then would be CGU, our third UCG. And notice how that completely changes the reading frame as compared to when we were incorrectly reading it. So AUG dictates how those triplicates will line up. So you could go through this now and determine what amino acids would get incorporated, knowing that of course FMET is the first amino acid at our most amino terminus. And if you're up for some fun, pause the vodcast right now and see what these next triplicates would tell you. What would the second amino acid be? What would the third amino acid be? And then finally the fourth. We'll do a little more of that practicing in just a minute. Okay, so now that we've looked at how to specify the reading frame, let's make sure that we understand the major players in all of this. That is, we need to introduce the ribosome, and we need to also introduce the tRNA. So ribosomes, as I've said before, and I'll probably say again, are kind of the most misunderstood of all cellular structures, I believe, because they've so often been called the workbenches of translation, making them seem totally static and benign. And that's not at all what they are. In fact, they're quite the opposite. They're very active in the process of translation. And in fact, it is some of the RNAs within the ribosomes that literally build and form the polypeptide chain. They synthesize the peptide bonds. So I like to call the ribosomes instead the translation factories to indicate their really active role in the process of making the polypeptide chain. And we know that they move along that mRNA in the 5' to 3' direction. That is, they read the mRNA in that way. Each time they come across a codon, they're going to then play that role in helping to facilitate the incorporation of the next amino acid that's specified by that codon. So they do facilitate the joining of the amino acids, and in fact, it's specifically one of the components of the large ribosomal subunit called the 23S ribosomal RNA that literally makes the bond, the peptide bond. And this is crazy because what we're seeing with that is that this RNA, this 23S RNA, is acting like an enzyme. So even though it's a completely different kind of macromolecule, it's an RNA and not a protein, it's acting like an enzyme. Those are actually called ribozymes. And if you're feeling like dorking out with some of that, which I bet some of you probably are, go and check out this lecture that I've listed at this URL by a man named Thomas Cech, who worked at the University of Colorado. And he really was the person to lay the foundations for research on RNAs that act as enzymes. It's wicked cool. So if you have a chance, you should definitely look at that lecture. So there may be as many as 15,000 to 20,000 ribosomes in an actively growing E. coli cell at any given time. So think about, it boggles the mind to think about the massive amounts of protein synthesis that may be happening in an E. coli cell at any given moment. Now let's dissect the components of the ribosome, remembering that it's only when the ribosome synthesizes protein that both the 50S and the 30S subunit come together to form the full 70S ribosome. But if we look at each component, the 50 and the 70S, you'll notice that the 50S subunit is composed of two RNAs, the 5S and the 23S RNA. And remember, the 23S is the one that we mentioned is sort of the shiz-niz of all of this. It is building and forming the peptide bond when synthesis is occurring. Of course, there are also many polypeptide chains in this because we know that the ribosome is composed of two macromolecules, both RNAs and proteins. So we'll see that polypeptide chains are also an important part of this, in fact, 34 of them in the large ribosomal subunit. The small ribosomal subunit, the 30S, is also made up of a very famous rRNA, the 16S rRNA, which in order to identify and classify bacteria, the 16S rRNA is commonly sequenced. In fact, there are huge databases, if you guys have heard of GenBank, out there that have just cataloged 16S ribosomal RNA data just to compare organisms with regard to their similarities. We also recognize there are 21 polypeptide chains inside of the small ribosomal subunit. So the ribosome's a super important component of all of this protein synthesis. In fact, they very much are the factories of this process.